Can you hear me okay? Okay, I hope you can hear me. So today we will talk about, or we will start a series of lectures around the thyroid gland. And this will complement some of the things Carlos has talked to you about, and will lead us into the adrenal gland. So by the end of the totality of these lectures, I would like you to be able to describe the gross anatomy of the thyroid gland, explain how it's regulated, and regulation of its role in the hypothalamic pituitary thyroid axis is involved, which hormones regulate it, which hormones are produced, and then understand the feedback mechanisms. I would like you to be able to describe and discuss the synthesis, secretion, and functions of the thyroid hormones. Be able to explain the pathophysiology of thyroid hormone imbalance, which has wide-ranging effects on physiology. And I would like you to be able to describe the presentations, investigations, and treatments of hypo and hyperthyroidism. So we'll start with just explaining the anatomy of the thyroid gland, and then we'll move on to synthesis. So this is a rough macroscopic viewpoint of the thyroid gland. It was recognized early on anatomically because it has this butterfly shape, and it has two halves on either side. And the two halves are called lobes. You have a left lobe and a right lobe. And down the middle is a narrow piece of land, or a narrow piece of the thyroid gland called an isthmus. And this, we say an isthmus in English, is a narrow piece of land connecting two larger areas across an expanse of water that otherwise separates them. We have two lobes, left and right, joined by an isthmus. In terms of size, the thyroid gland weighs roughly 30 grams in men, and it's one of the largest endocrine glands. In terms of development, when it's formed about 4 weeks after conception, and by 12 weeks of gestation, the gland is synthesizing and secreting thyroid hormones under the fetal HP axis. The thyroid gland, as you can appreciate, if it's formed so early, is essential for normal growth and development of the fetus. As you can see also from this image, it is highly vascularized, okay? Which means the delivery of the hormones, right, as its endocrine function, is easily facilitated. With blood flow going through around 4 to 6 milliliters per minute. So quite a bit of blood flow, lots of vasculature, lots of exchange of endocrine hormones. So the thyroid is a very important process in development and in homeostasis of our physiology. So what does it look like under the microscope? So this is just a whole section viewed with low power under the microscope, cross section. And you can see here it's formed of this lumen of the trachea. And then next to the trachea, you can see the thyroid, that butterfly formation. Okay? And if you look very closely, you can start to see little circles, which I will show you briefly what those are and what their role is. So the functional unit of the thyroid is something called the follicle, and that's those circular things I was showing you. And the follicle is made up of a single layer of cuboidal cells surrounding a clear mucus-like fluid called the colloid. Sorry, my image is hiding here. You have the colloid inside, and you have these cells just around it, okay? Cuboidal cells. And then inside, you've got mainly this glycoprotein called thyroglobulin. We'll explain how thyroglobulin works, but it acts basically as a sink or as a reservoir for thyroid hormones. And then in between, you've got blood capillaries, and you've got these parafollicular C cells filling in the space in between the colloids. So it has a very distinct and unique look about it under the microscope. So this is a view, a little higher power view under the microscope. And you can see very nicely the colloid. You can see the blood vessels. And you see the follicles. So if I were to show you that, you should be able to identify those distinct features and tell me that we're looking at a thyroid gland. Now, those C cells that we mentioned that were kind of in between the space are responsible for synthesizing and secreting calcitonin, and an important hormone which helps regulate plasma calcium. And you may have heard recently the calcitonin receptor was a target for treating severe migraines. And the structure came out last year. C cells are also called parafollicular cells and are derived from the neural crest. They occur singly amongst the follicle cells, or more usually found in small groups between the follicles. You can identify them because they're outside the circle, outside the follicles. You can recognize the fact that they're outside because this term parafollicular, para in Greek, ancient Greek, means besides or next to, near, contrary to. So we have the follicle and the parafollicular cells. So again, single cells of follicular cells forming these circles. Inside you have this colloid filled with follicle, which is we'll go into thyroglobulin hormone. In between the follicles, you have parafollicular cells and blood vessels, along with this white connective tissue to maintain the structure of the entire gland. So that is the gross anatomy of the thyroid. Now, what does it actually do, and what are these hormones that it makes? So let's talk a little bit about the synthesis of the hormones, because they're directly related to also the structure or the anatomy. So these are the major hormones that we talk about when we talk about the thyroid gland. We commonly refer to them as T3 or T4. We will sometimes mention thyroxine. In general, I will refer to T3 or T4. So the primary hormone secreted by the gland is T3 and T4. Sometimes you'll also hear them called iodothionines, and then the calcitonin that I mentioned to you. Just again to remind you that the calcitonin is secreted by the C cells, so different cells, independently of the other thyroid hormones. So although they're next to each other, they don't work in conjunction. So I'm not going to say much more about calcitonin itself, other than it is another hormone secreted by the thyroid gland, and it's made by these C cells. That was all I expect you to remember. However, we will talk quite a bit about T3 and T4 and how they're made, and I will expect you to know the details. So, thyroglobulin are large glycoproteins, over 600 kilodaltons, and they're secreted by the follicular cells into the lumen containing colloid. And the structure of the peptide backbone of thyroglobulin looks, you can see here, okay? You can recognize the tyrosine backbone, right? And later inside the lysosomes of the follicular cell, enzymes will cleave the two peptide bonds, releasing T4. So each molecule of thyroglobulin contains over 100 residues, which can be iodinated. Okay, so this is the backbone here of thyroglobulin. It has over 100 of these, which can then be iodinated. So the process is like this. First, you start with a tyrosine, then it gets iodinated by a thyroid enzyme called thyroxine peroxidase to form monoiodotyrosine, or MIT. And then you can get a second addition by the same enzyme to form diiodotyrosine, okay? And then you can either have addition, so this occurs, this enzyme will make either MIT or DIT, so monoiodotyrosine or diiodotyrosine. And then from those, you can then make your T3 via conjugation. You conjugate a MIT and a TIT together, and then you have T3, or you conjugate two DITs together, and you have a T4. So So these are the two forms that are then secreted, okay? These T3 and T4 are released only after proteases from the lysosome cleave them from the TG, okay? So if we go back to what the TG looks like, you have to get cleavage from the TG to release T3 or T4, right? Here's the bonds here in red showing you these peptides that cleave T3 or T4. And then this here would be either T3 or T4 depending on the form of iodotyrosines that are there, okay? Obviously in the previous one I was showing you T4. Okay, so step one to make this thyroid gland is to first bring in the iodide, because as you can appreciate, that is the key step or key aspect or the key ingredient, if we should say, of the thyroid gland. So how do we bring in iodide? What's the trick? And what's the trigger for bringing in iodine? Well, first, we have input from thyroid stimulating hormone, okay? This acts by a thyroid stimulating hormone receptor, which is a GPCR. It leads to the production of cyclic AMP and all those steps we learned about in the first lectures. It says my video is slow, guys, so I'll turn off my my video. Let's see if it maintains. Let's see. So TSH binds to TSH receptor. You then trigger cyclic AMP production. Okay. This cyclic AMP then leads to activation of a sodium iodine pump. Okay, here. This pump will then actively trap iodide from the plasma. Okay. And then transport it across the basolateral membrane. And now we've brought in iodide into our epithelial cell. This iodide, because it's a small molecule, can then diffuse across the follicular cell membrane into the apical membrane and into the colloid, okay? So, thyroid stimulating hormone binds to our thyroid stimulating hormone receptor. This stimulates cyclic AMP via our GS, heterotrimeric G protein. Cyclic AMP is produced by the adenylate cyclase enzyme. As cyclic AMP levels increase, this activates our iodine pump and allows iodine to diffuse, or enter the membrane, enter the cell, and then diffuse across and enter into the colloid. Okay? Now, obviously, how does the iodide get there? Well, remember we said this organ was highly vascularized. So you can see here the iodine can enter very close by the bloodstream, and then this pump just brings it in from the outside. Now, what happens after we trap our iodide molecule? Well, the first thing we have to do is we have to oxidize iodide to iodine. And this is done using hydrogen peroxide and an important enzyme that's in the thyroid called Thyroperoxidase, or TPO. So TPO takes hydrogen peroxide that is located within the colloid and oxidizes our iodide into iodine. This does two things. One, it prepares the reaction for addition to our thyroglobulin, and it also serves to trap the iodine within the colloid, as the iodine is less likely to cross back over the membrane. This process then allows iodine to be used in the next reaction, okay, where we have iodination of the tyrosine groups on the thyroglobulin. This forms the mono or diiodotyrosine that we talked about, and then this serves as a giant reservoir for storing your hormones. Okay, you can think of it as a long train if you want. And you know, in a long train, you take the cars off one at a time. Imagine a train traveling from Shanghai to Nanchang, a very, very long train with lots of cars, and eventually, as you pass, right, each province, you'll take off a few cars as you need them, and this is very similar for thyroglobulin. Gradually, you get conjugation of MITs and DITs, as we talked about, forming different T4 or T3 components. And this prepares them for later cleavage by those lysosomal enzymes that we discussed. Now, the next step involves what is called retrieval. So we still have the thyroglobulin on the wrong side, so to speak. It's located only in the colloid. So we've got to get T3 or T4 back across, right, so that it can be put back into the bloodstream and released on demand. So how does this happen? So thyroglobulin is taken into the follicular cell via endocytosis, which you will remember from your cell biology. These vesicles, termed specifically colloid droplets, fuse with the lysosome, and the pH change from the endosomes to the lysosomes activates those proteases, which then release T3 and T4 via proteolytic cleavage of those peptide bonds. This then frees T3 and T4, which will now be secreted into the plasma membrane. Sorry, into the plasma, the surrounding blood vessel that we talked about. Okay, so we've got to get our thyroglobulin across. To do that, we have endocytosis. From endosomes to lysosomes, the pH change activates the enzymes. You get peptide cleavage of T3 and T4, and these can now move across the membrane into the bloodstream. Importantly, the thyroid is very efficient machinery, and it doesn't waste anything. So it takes the MIT and the DIT that were used previously, recycles them using by deiodinase, so it removes the iodine groups that were not secreted, and it allows recycling of iodine. Importantly, 95% of the THs leaving the thyroid gland is in the form of T4, thyroxine. And this typically functions as a circulating prohormone. So what is a prohormone? It means it's not very active and usually processed one more time to be the more active form. Prohormones are typically used because they're more stable in the plasma. And that is indeed the case in this situation. So, the majority of the hormone that leaves the thyroid is T4. Then within the target tissue, there's another enzyme, this deiodinase enzyme, which converts T4 to either T3 or something called reverse T3. Reverse T3 is completely inactive, and T3 is the more active component of a thyroid hormone. So T4 is secreted by the thyroid as a prohormone. Then you get in the local tissue another enzyme called deiodinase. This deiodinase then converts to either T3, the active form, or RT3, inactive. Just to give you a sense of how active T3 is, it is about 40 times greater than T4, and RT3, the reverse T3, is biologically inactive. 90% of the biologically active thyroid hormone within the cell is in the form of this T3, okay, so the active T3. Now remember, I mentioned to you, why do we use a prohormone? What's the purpose of it? Well, it's because the half-life of T4 is 6 to 8 days, while that of T3 is only 1 day. So immediately you can appreciate the importance of having a prohormone. The only problem with this prohormone is going to be exemplified in the next few slides, where it's the this idea that the prohormone has such a long plasma half-life becomes a problem when you start talking about nuclear accidents. So many of you were probably too young to know or hear about. I was only a young child myself. Something called the Chernobyl Accident that happened between Ukraine and Belarus in the 1980s. And it was the result of a flawed reactor design. Okay, so it was an engineering failure. And the engineers responsible did not respond in time when the reaction, for those of you that do know that nuclear reactions are controlled reactions, and if it can grow out of control, it can lead to an explosion. So it wasn't dealt with, and the resulting steam, explosion, and fire released about 5% of the radioactive reactor core into the atmosphere. And this was about 5,000 PBQs, which is a measurement of iodide, of radioactivity. It was I-131. So two workers at the Chernobyl plant that were died on the night in the accident, and 28 people died within a few weeks as a result of acute radiation poisoning. Okay. Now, unfortunately, at the time, there was no evidence of a major public health impact attributable to radiation exposure 20 years after the accident. But the reality is that was a bit inaccurate, and it was only appreciated later that how serious the accident was and the impact on the population. Okay. First, let's just talk about the environmental impact. You had 70% of the radioactive substances blew north over the population of Belarus. A quarter of the country's best farmlands and forests were poisoned for tens of decades by both cesium and strontium, and plutonium became within the land, within the earth. Belarus had to evacuate hundreds of towns and villages, and thyroid cancer in children increased by more than 100 times due to the large amounts of radioactive iodine that they have ingested. And the problem was, I just explained to you how iodine was trapped, right? And the minute it was brought in, okay, and discovered that this half-life of 8 days, right, for the prohormone, it quickly was understood, and it was appreciated by someone called Glenn Seaborg and John Livingood, discovered in 1938 that the half-life and radioactive decay of iodine was about 8 days. So that's about the half-life, remember, of that T4. Now, those guys discovered it when they were looking at nuclear bombs for World War II, and this was only appreciated later with Fukushima nuclear accident, which happened in Japan. So the problem with the prohormone, T4, and the half-life is that iodide from the environment, the radioactive iodide, was taken up via all these routes, okay? So you had iodide entering into, and iodide was extremely stable, right? With a half-life of 8 days, which means even in 2-3 weeks, you still had quite a bit of iodide still around, and can enter into all these different areas of the food chain and very quickly get and all isolate into our thyroid here, right? And that's the problem with using radioactivity. And now there's an important children's thyroid cancer treatment center located not too far from Chernobyl to try and deal with all the poor children who were implicated in that accident, which was really just a terrible story. And it explains the importance of, for us, of using environmentally friendly energy sources, right? And taking care of our environment so we can all be healthy and have healthy children. So, okay, let's take a short break, and then we can get back and cover a little bit more about and start talking about some of the diseases associated with the thyroid gland.